The overall goal of the following experiment is to assemble and characterize a small interfering RNA or siRNA delivery system. This is achieved by grafting porous silicon microparticles with polyethylenimine and arginine to create a platform with a cationic charge. As a second step, siRNA molecules are mixed with the microparticles, which results in siRNA binding. Next, arginine polyethylenimine siRNA nanoparticles will be formed as the silicon gradually degrades in order to enable sustained release of the siRNA.
This technique could have implications for the treatment of various diseases. It enables the efficient delivery of small interfering RNA.
First, oxidize non-functionalized porous silicon particles in a 30% solution of hydrogen peroxide at 95 degrees Celsius for 2 hours. When finished, aminate the oxidized particles in a 2% 3-aminopropyl triethoxysilane solution in isopropyl alcohol for 2 days at 65 degrees Celsius with gentle stirring. Transfer the particles to a centrifuge tube and centrifuge the solution for 30 minutes at 18,800 times G. Following centrifugation, resuspend the pellet in isopropyl alcohol using brief sonication. After repeating the centrifugation and washing step with isopropyl alcohol and ethanol, leave the particles in ethanol until ready to use. Next, add 10 microliters of particle suspension to 10 milliliters of isotone diluent and count the particles with a particle counting analyzer to determine